Yeah,
so you play guitar?
I tried to learn for a while, but didn't get the dopamine that I needed.
Sex and drugs and rock and roll.
Banana milk, that's what you want.
I should have, used artificial dopamine.
I did some of that.
That's what Eric Clapton did, right?
This is the Convergent Science Network podcast. Leading researchers in the domain of neuroscience, brain theory, and technology are interviewed by Paul Verschure and Tony Prescott.
This is Paul Verschure with a Convergent Science Network podcast with my colleague Tony Prescott. And we're talking with Gregg Recanzone, who was speaking today at the BCBT Summer School 2015 here in Barcelona, on something that I was told as a student didn't exist, which was adult cortical plasticity. So, Gregg, why did you go for that topic? What's important about that question?
Well, the part of the reason why I did this, that I talked about this, which I don't usually talk about when I give seminars, is that Leah specifically asked me to, and she reasoned that back in the days when we were graduate students, it was, as you said, impossible, and it didn't occur, and it was a real revolution in the way that we thought about how the cerebral cortex worked. And it's been so well accepted at this point that students today just kind of take it for granted and don't really get ever taught kind of the classic papers and studies that were done in order to demonstrate it to the point where we all now, believe it. So that was my main motivation today. Mm-hmm. And plus, it's 8 years of my life working on that thing. So it's kind of nice to revisit it and still see some enthusiasm about that kind of stuff.
Right. Yeah. But now, how do you explain that transition historically?
That's a good question, too. It seemed, as I was going through the beginning of my talk, that back in the 1800s, everybody knew that it had to exist and it was trivial. And then for some reason, since nobody had been able to really demonstrate how the brain changed when you actually did learn something, and then Hubel and Wiesel said, look, it stops changing after a certain period in development, which made perfect sense. It just got kind of lost in that shuffle. And so it took a 10-year hiatus, really, where people just kind of thought critical period, and nothing about, wait a minute, how did I just learn about this? I'm an adult. My brain had to have changed somehow, right, for me to learn this, but it couldn't have, but it just, I have no idea why people got stuck on that. Mm-hmm. And coming from Merznick's lab, where he never considered that as a serious proposition for learning, I never got caught up in that mindset, right? So I always believe your mentor. That's always important, right?
I recommend exactly the opposite to my students. But then I add to that, disagree with them, but give a better alternative.
Yeah, absolutely. Okay. Yeah.
But now, for you, this history starts with Mike Merznick and John Kaas,
right?
Very much, right? So what was the key insight you think that brought them to that point of reviving these ideas about adult plasticity in the cortex?
Well, I've talked to both of them about this a number of times, and because it was remarkable, it was a stretch, and it was a struggle, because many people did not want to believe those early papers, the digit amputation paper and the median nerve section. They wanted to think of unmasking and all these other things, but they're both pretty passionate scientists, and they both thought that it just made more sense that it worked that way, right? And I think they just believed, right? And Mike would say that all the time, that he just kind of believed that it had to be something like that, and he just wanted to show how it was done. And so those early studies I thought were brilliant in the sense that they weren't supposed to work, and every time they did something, they learned something new. And so it was a really exciting time for everybody in those labs to be doing those kinds of experiments.
So can you describe one of these sort of early experiments that really brought the message home?
Right. Well, for me, the one that actually solidified in my mind what happened is when you look at the map of the hand in area 3B in a monkey, which is really precise and elegant, and each finger is represented very cleanly and distinctly, and from the wrist to the fingertips, it's all very topographic, right? And then the critical period genetic model would say that if you lost a finger, then that part of the brain just wouldn't have anything to respond to anymore, right? And what they did is ask the question, is that really what happens? And so what they found was that indeed the neurons that used to respond to the missing finger now responded to the other ones. And another key insight that I didn't talk about today that really made them think about it differently is that it wasn't simply the case that the old map was there and then some new things happened also. The whole map looked like a four-fingered monkey. So there was the same kind of overlap and the same progression and the same topography, but just with the missing finger. And so that gave them the insights that what must be happening is that the receptive field of a particular neuron has to be dynamic. It has to be able to change over a very short time course, right? Probably, in a dramatic thing like this over the course of 2 months, but probably does it over the course of several days, right? Mm-hmm. And so there was something in there that these neurons are always actively changing the weights of their synapses to see what parts of the skin that they should represent relative to what all their neighboring neurons are representing, right? So and that's how you keep the topography. Now, it was really lucky they did that in area 3B, because if you look in the visual cortex that Charlie Gilbert did, it doesn't work that way. They all stack up, right? And so it's not this ordered topography that you see in the somatosensory cortex. In the auditory cortex, if you do a cochlear lesion, essentially what happens is the cells that used to respond to the now lesion frequencies respond to what the one that was spared, the edge of the lesion. So all three of them are very different. And so it was really fortunate that they did the 3B study because it made the most sense, right, at the time.
But also in retrospect, it was the most effective choice.
Yeah, in retrospect, they didn't know about the other two, right? Those came subsequently. So they lucked out, essentially. Right?
But now, when these amputation studies were done with these results, what was the resistance that you faced in the community?
Yeah, so personally, fortunately, I didn't face any of that. So Mike was a champion. He would accept anybody's invitation to go out and talk to their research group and give a spiel, and then spend the next half hour defending it because people said, no, you can't change the brain like that. It's just unmasking, or you just, it's just something that's not in any way related to any sort of perceptual differences, right? And I didn't see him a whole lot as a graduate student because he seemed to be gone all the time, going out there and pitching his story and defending it the way that only he can do. And eventually people came around, I guess. I mean, when the accumulation of evidence, if he had stopped after the digit amputation, it would have been all over. That would have been the end of it, right? But we kept going and going and going. Then other people get on board too and start doing these experiments. And it's like, look, it always changes, right? So it must be real, right?
So what is the now consensus? So, because obviously we have a revised view of plasticity. This battle has been won, but also the notion of critical periods is still there.
Sure.
We've talked about it several times in the last week. What would you see as the consensus now about the amount of adult plasticity and how that compares to plasticity in infancy?
Well, there's absolutely no doubt that there's critical periods, and the lid sutures do cause ocular dominance column shifts and all these kinds of things. So that's absolutely certain, right? The way I think the field has come to a consensus is you have these developmental time points where you can do largely anatomical differences, right? But not necessarily. And then once all those things are over with, you're left with the adult plasticity, which is how you change the weights of your synapses and how you mess with inhibition and things like that. And that is what's basically the difference between critical periods and adult plasticity is in adult plasticity, you don't expect much, if any, changes in the underlying anatomy.
So there's fundamentally different mechanisms.
Yeah. So it's fundamentally different. And they're both working independently, essentially. Yeah.
So now we look really at changing the periphery. We remove a finger, or you suture two fingers together, so you make a four-fingered monkey, and then you see these changes in the somatosensory presentation of the body as you describe. But now, how far do these changes percolate into the system? Like, for instance, if I have a somatosensory change due to this change of my hand, is that matched also to changes in, let's say, primary motor cortex, premotor cortex? How far do these changes really percolate down into the system?
That is a question that, to my knowledge, hasn't ever been looked at strongly. So I know that Randy Nudo did a whole series of experiments where he had the monkeys do different sorts of motor tasks, and then you look at the motor cortex, and there's predictable kinds of changes that would associate with that. And so clearly, anytime you're interconnected networks like 3B and 3A and M1, you're going to see these changes percolate, at least to the extent that they need to be, right? So I imagine that if I looked in the motor cortex of those same animals that I studied, I would see all kinds of things that were related to the reach and to the release and all these things because they were doing that, and that was very important to them too, right? So is that because of the somatosensory changes? Probably not as much as it is to the actual motor movement, but they're going to work together. But auditory cortex didn't change, even though they heard the stuff, right? So there's some limit to how much it goes, and it's probably just to what's really primarily task related.
Mm-hmm. Yeah, because that was sort of the next step in your experiments, right? So now we first change the periphery. You could say, okay, if you want it's a rather strong perturbation, so we have a reorganization of the map. But then you start to look really at the learning component of this by engineering a task where you provide haptic feedback or tactile feedback to the monkey, and then trying to see what kind of changes you would then get in the somatosensory cortex. So, what kind of changes did you get there in that experiment?
The changes in the somatosensory. Yeah. Okay, so the monkeys were trained to feel a vibration on one small part of their finger, starting at 20 hertz, and to detect when it went faster in a subsequent presentation. So we saw a great deal of changes, some of which were related to the behavior and many of which were not. Okay, so one thing that wasn't related to the behavior, as far as we could tell, was the size of the receptive fields got bigger. And the thinking at the time then was that, well, that's because it's a pretty big stimulus. It vibrated a lot of skin, and so everything is being synchronously activated by this 20 hertz stimulus. And that's a consequence of the mechanisms by which you change your weights, right? Another thing that happened there was a expansion of the representation of that skin, right? And that was correlated with the ability of the monkey to do the task, but not super well. And if that had been the end of the day, I would have been perfectly happy with it because that's what I was looking for, a change in the map related in some way to the change in the behavior. The main thing that was related to the behavior was the ability of the neurons to follow each individual cycle of that 20 hertz or 21 or 24 hertz stimulus. And what was interesting to me, the two changes that really made the big difference was for the brain, when the skin was stimulated on the finger that was being trained, there was a lot more neurons that responded in that way to each cycle of the stimulus. But every individual neuron that did respond that way responded about as well as ones that did the untrained skin. Okay, so if you did some sort of measurement of the fidelity of the response relative to the stimulus itself, there was no difference between trained and untrained. Okay. And so that would mean, well, the signal is just louder because there's more neurons doing it. But it was more than that. What happened that really made the difference was for the untrained skin, or normal skin, if you will, the way that the timing of the response to each cycle of the stimulus varied quite a bit between individual neurons. Okay, so that would make if you did all the population, you have a broad tuning function. In the trained monkeys, they all lined up with each other. Okay, so the temporal fidelity across the population was enhanced. So it was a louder signal, it was a better signal. Okay, so it got much tighter, which allowed the monkey, I'm interpreting, to be better able to tell the difference between 20 and 22 hertz because there was less slop, less variability, and it was a much cleaner, tighter signal.
Right? But you would say, is that expressing something like what fires together, wires together? Like I'm driving these neurons simultaneously, they fire simultaneously, this drives local plasticity, they get coupled more tightly together, and as a result, their response becomes sharper in time.
Absolutely.
Okay.
Yeah.
But then the other thing you showed is all this elongation of these receptive fields.
Right.
That's the thing that didn't make any sense to me. Why would these receptive fields get so much larger in terms of their sensitivity along the finger?
Along the finger. Yeah, so when the monkey has his hands on the apparatus and the thing comes up and starts to vibrate, it vibrated quite a bit, right? And so it wasn't the same as when you're doing the mapping experiment and you're just barely touching the skin. It's actually a pretty significant stimulus. And I felt it. I wanted to see what it felt like. And it felt pretty big. right? So it was as though, even though the probe was kind of small, since it was moving so much, it was moving a lot of skin, almost the whole phalanx. And I think that's wires together is the same kind of thing. You're moving all the skin together at once, right? And there was no penalty for having a big receptive field, right? I would guess that if I had 2 probes and I asked the monkey to tell me, am I just doing 1 or 2, then they would get small.
But it needs more than wires together, because there's an effective reinforcement here. You need the reinforcement.
Right, exactly. Yeah. They have to know that they're doing it right, and it's
this making this discrimination gets you the peanut, whatever you want.
Exactly.
Yeah. So it's a more complicated system than simply Hebbian learning. If you experience something a lot, then through Hebbian learning, you might refine your receptors. But if it's important for a reward, then that's going to be, I think, as you've shown, much bigger effect.
So if you do exactly the same stimulation, but the monkey listens to the sounds instead, then you don't see the big receptive fields and the big area and the tighter tuning.
So this is not Hebbian then, surely. It's or at least it's a Hebbian rule that's being modulated.
Well, that's the question, right?
Yeah.
Is it just driven by the statistics of the input, or does it depend on an additional neuromodulatory signal that says, this is really great, we should get it again?
Yeah, I think it's the latter.
Okay.
Yeah.
Mm-hmm. So, but now the other thing you saw is also in terms of the response latency, there were changes, right? The neurons also seem to be responding earlier to the stimulus.
They were earlier,
than the control condition.
Mm-hmm.
So how would you counter that effect?
That's a really good question too. Why would you get, I mean, it's clear to see why you would get sharper, right? But why would you get sharper and earlier too? And I have no really good answer for that, right? So it's somehow as though these early responses are the ones that the monkey was using and pulling the other neurons towards the shorter latency.
Okay.
Right?
But it was a discrimination task that the monkey had to perform.
Right.
So was sort of you changed the stimulation frequency, and the monkey had to say, I detected that. Right. If it was correct, it got a reward.
Right.
So how frequent were these changes in frequency?
So the way we decided to do it is the first one was always 20 hertz. If I remember correctly, the second one was always 20 hertz, and then it could change on the 3rd, 4th, 5th, 6th, or 7th with equal probability, right? So in average, there was 5-ish of these 600 millisecond duration things. So we were thinking at the time that we had to do a lot of stimulation because the Jenkins experiment had millions of stimuli and saw big changes. So I was afraid that if you didn't do quite so many, right? If you did just, one and then another one, 2 choice, right? Then that wouldn't necessarily do it. I think now that would have worked and probably would have saved me a lot of time.
Mm-hmm.
But
right. So it means, well, looking across these 2 experiments, you see that differentiation leads to changes in the map that probably have a lot to do just with the input statistics to the map, because this is not task dependent in any way.
Right.
So when we start to bring in a task, and it's really very much also the reward or the task contingency, the relation to reward or punishment, that would then impose further changes to the map. You agree with that interpretation? Then
yes.
2 learning mechanisms at work. Okay, but now how stable would that learning be? Like, for instance, if what we already saw, what these peripheral changes, like fusing 2 fingers and then disconnecting them, actually the map follows that manipulation. But it might do it more slowly, as it might follow, let's say, the task contingency in the stimulation experiment you described.
Uh-huh.
Right. So what is sort of the characteristic time constants of these 2 learning processes?
Yeah, that's something that I never address specifically, but it's obviously a critical component, mostly because the idea, of course, is that you can use this for learning for good, for rehabilitation, and all these kinds of things. And how long does it take to do this? And once you do change, do you ever want to change back, or can you change back, or do you need to change back? And so I think that's going to be really largely task dependent, and really largely how much neuromodulator can you get in there based on pharmaceutical, or just the desire to do it, or the importance of the stimulus to you, et cetera. So my guess is that it will probably take days or weeks or months, depending on all those different kinds of things. right?
Okay, but in case of the differentiation or the amputation experiment, how much time does it take for that map to stabilize?
Yeah, unknown. Okay. So those early experiments, they also did 2-digit amputations, and they waited 2 months or 3 months or whatever. And what they found there was that there would be a silent zone. Mm-hmm. So the 2-digit amputation deafferented too big of a portion of the cortex for the afferents to actually reach each other. Then Ted Jones and Tim Pons looked at the Silver Spring monkeys a decade or two later that had been had dorsal rhizotomies done to them 15 or 20 years beforehand, and they saw complete recovery, right, in the map. And that was correlated with changes in anatomical distributions as well, all the way from the cunea and the thalamus up to the cortex. So if you wait long enough, presumably, that was a huge deafferentation. That's the whole arm, right? Yeah. And they still saw centimeters of change as opposed to, I think the early studies were something like 500 or 600 microns. If you got beyond that, you couldn't recover or you couldn't change your map. But, you know, you only wait 2 months, right? If you're not going to really wait 10 years, that's a hard grant to get in the United States.
Sure. but sort of the task-dependent change under the influence of neuromodulation can go relatively rapid, right?
Right. Months, yeah. Days or weeks, yeah.
No, with a task-specific change, certainly if you go into the middle of conditioning, you might see map changes in dozens of trials. No, it might really quickly.
Norm Weinberger has done that for decades.
Exactly.
Yeah.
Right.
Yeah.
Well, then these amputation experiments show that the overall, let's say, topological organization of the map dependent on the periphery. It might be a much slower process that might require, let's say, throwing out new processes, building new connections, and so on. Would you see it like this? That the amputation, the processes underlying the reorganization after amputation are slow, and the task-specific ones, depending on neuromodulation, are fast, or you wouldn't really see it in those simple
I would
terms?
say that's probably accurate, because when you think about a small deafferentation, right, the consequences of that to the animal are not as dire as you have to do this task in order to get your food, right? So how much neuromodulation would there be in a situation where you have a digit amputation? It's probably not nearly as much. So in that sense, it would go much slower because the need for it to go faster is just not there.
You mentioned in the talk, you were talking about this as requiring attention. So what's interesting is attention doesn't necessarily imply reinforcement. It implies some maybe more intrinsic motivation to attend. I guess you're not directly getting at it in this task, but for instance, in the case of a person who's learning some motor skill, their reinforcement might be, you might have to wait a long time for it, but you're intrinsically motivated. So we don't necessarily have to think about, maybe we do want to think about a burst neuromodulator, but the system that's delivering this could be some really quite complex system around intrinsic motivation signals rather than obvious rewards.
Right. So imagine learning to play guitar, right? Which is difficult to do, I can say from personal experience. And the motivation is to sound good, right? And that's very esoteric, right? So now your auditory cortex is deciding how much attention a neuromodulator your motor system is going to get, and your tactile system based on, your desire for whatever that is to sound like you're not a hack, right? To sound good. So, yeah.
And you don't necessarily know what the task is. I mean, it's... you don't know what you need to do to sound good. You're exploring the space. There's a lot of trial and error.
Right.
But I guess you're getting at the basis for that here. So.
Right.
But do we really need to have an attentional interpretation of this? I mean, if you just get the banana chow, whatever you give these animals as a reward, driving in neuromodulators like dopamine, which then modulates plasticity in the cortex, leading to this reorganization, do we need to say, and we paid attention to it? Do we really need that interpretation? It cannot just be task contingency.
It could be task contingency. It could be reward, dopamine. It could be norepinephrine. It could be all kinds of different things. So to use the word attention, as I did in my talk, was a pretty loose interpretation of the people who'd study attention, right? So they would shy away from that necessarily being
Right.
the key, right?
Mm-hmm.
I kind of throw the word out there just because right after I left Mike's lab, Mike Kilgard came and he essentially did the auditory version of the experiments in rats. And instead of training them, he just electrically stimulated the basal forebrain and got all the same effects.
Right. Exactly.
So, and there was no attention there, right?
Right. But that was auditory conditioning, right?
Right.
Exactly.
Yeah,
exactly.
Yeah. So he dumps a bunch of acetylcholine on the cortex right when the sounds are playing, and the maps change like crazy.
Right. Yeah, exactly.
Yeah.
So after these experiments on that really demonstrate, that really contributed significantly to demonstrating that we have adult plasticity in the cortex and to show also some of its organizing principles, in particular the role of neuromodulators, I think, and let's say input statistics on how these maps are organized. What are the boundaries on that process, though? I mean, would you be able, if I would give you just enough time and monkeys and training equipment, could you just easily retrain the auditory cortex into a visual cortex and make it as large or small as you wanted it to be?
In adults.
Yeah.
Take normal adults and then see how you can manipulate that. Yeah. So we heard a couple of days ago in the seminar that you blindfold a person and their visual cortex will start helping them learn Braille, right? So in that sense, you know,
or to echolocate.
Exactly. So in that sense, all bets are off. It sounded like from that limited amount of data, right?
But where would you start when we're going to make an echolocating monkey? Where would you start? How would you do this most efficiently?
Probably be easier to teach him to read Braille than to echolocate, at least from the training point of view. And then if you were going to do that, if you deprived a bunch of cortex of its normal input, then it would and demanded that it use its hands, efficiently well, and its visual cortex, for example, is not doing much of anything. I think it would take days to weeks to start getting that activated without not doing the neonatal manipulations that you can do to get all this rewiring that Marganka showed a long time ago.
Right.
So yeah, in 1992, when those publications came out, I'd say, well, that's never going to happen because there's no substrate for that, right? It's just not going to work. It's all cortical and it's all done, right? Today, we know much more about it. And so it is possible to do these kinds of things. And exactly how that's done is still not entirely clear, but, it happens. So you can change these things.
So the second part of the talk, you focused on sound localization, right? So we moved sort of to different preparation. We moved also to different kind of tasks.
Right.
So why does sound localization help you to understand this issue of adult plasticity?
Right. So I started doing those sound localization experiments when I became an assistant professor, at least 5 or more years ago. And really my motivation at that time was to learn how the brain, what is it about the brain that allows us to perceive complicated things, right? And the plasticity part of my life, it seemed to me, was pretty much over by then. So I came, I conquered, I left, right? And I wanted to make a name for myself, independent of Mike Mersnick and Bob Wirtz, which is no easy task because these guys are just fantastic scientists, right? And so, like I said in the talk, there was really not much going on in the auditory cortex. And I think it's really, really important, right? So human beings, a lot of people will say, human beings are a visual animal, right? So we have this huge visual cortex and we just like to look at stuff, right? The other argument is that we're really a social animal, right? And what we really want to do is talk to each other. That's a strong motivator in the human brain, right? If you can't talk to other people, usually you go crazy. Right? You really need that sort of social interaction. So in that sense, we have this great auditory system that lets us do things like understand speech. And so in that sense, I thought, well, this is equally, if not more important than visual system, right? So it should get some people studying it. And we have the visual system to compare our studies to. So on the one hand, if you do the same kinds of studies that have been done decades ago in the visual system and the auditory system, it's new. Right? You already had this all sort of figured out if it works exactly the same way, right? So my motivation was to go ahead and do something that was novel. And what I really wanted to do was understand how complicated perceptions could be formed in the cerebral cortex. So people have said, oh, you were a somatosensory guy, and then you were an auditory guy, and you're a vision guy. I've always been a cortex guy. So I'm a cortical snob. I'll admit it. And I've always been interested in how does the cortex derive perceptions. Right? One way you can do it is you can have plasticity to get better. Another way you can do it is just say, well, how do you do it without a whole bunch of plasticity? How do you do this? And the auditory system and the sound localization system is really nice in my mind because it's not topographic and I know what it's trying to represent, which is azimuth and elevation. Mm-hmm. So I know exactly what the thing's trying to do, and that will give me some handle on figuring out how it does that.
Yeah.
Right.
So it not being topographic was a motivation you mentioned also in the talk,
right?
But isn't that make it life harder for you if you take something which is not topographic, or is the goal to show that the same principles apply whether there's topography or not?
Well, it does make it a lot harder, right? So if you're nicely topographic, you can do some really cool experiments like microstimulation or microlesions and things like this, and you can probe the system much more easily. I had tried for a while to do microstimulation of the auditory cortex to see if I could change the percept. Ken Britton is a colleague of mine at the Center for Neuroscience, and he has a long history of microstimulating MT and showing really elegantly that it does perturb the perceptual abilities of the animal. And he loaned me all his stimulation stuff, and nothing ever seemed to work. And I think it's likely because you can't just pick one spot like he could in MT with the best direction and set your stimulus up to do that. Instead, you stimulate this 1% of the neurons that are contributing to the percept, and you can never measure that in the noisy thing. So there's a distinct disadvantage to that. So the real motivation was thinking that a lot of the interesting parts of the brain that we do cognitive things in that we can't really study because we don't know anything about how those cells work, because they're not topographic and it's not an easy label. If we understood how you could do it in the cortex and something that you know is not topographic, then maybe you'd get some insights into how to look at other places in the brain.
Right. But no, you say you want to understand perception and complex perception, and then we will look at localization where it's sometimes you can say, well, all I need to do is just know the interoral time difference and the attenuation that I get through my pinna and my skull, and I have a good estimate of where things are, and I can just delegate this down to my superior colliculus.
Right.
So what's the leverage looking at sound localization? Actually, it is mostly studied looking at the superior colliculus. So what's the leverage you get looking at that problem at the cortical level?
Well, it's because the cortex is necessary for you to report to perceive where you've heard the sound from. So, for example, you can take a cat and you can put it and train it in a sound booth to go into the middle of the booth. You can play a sound from one of the speakers. It will orient towards the sound and walk towards it and get a treat. You leash in the cerebral cortex, A1 and around it, and then the cat doesn't do that. So the sound comes from the contralesional space. Sometimes it will move its pinna, sometimes it will move its head and face the speaker, and then it will walk to a random one. And what Hugh Hefner interpreted this to mean, it happens in monkeys too. He's done this in monkeys. And what he said is there is neural circuitry that tells you where the sound came from, collicular and brainstem, et cetera, but you don't have the percept that that's where the sound came. So when you're going to do something like walk toward where the sound was, you really have no conscious percept of where it came from, even if you're looking right at the speaker because you instinctively turned your head toward it.
So that means that the target setting is missing, or the transformation of identifying the target and location into a goal-oriented action is missing, or both?
At least the latter.
Okay.
Yeah.
So but then wouldn't you expect the deficit to not so much be affected by areas that are so close to auditory processing, but more those that are close to working memory, goal setting, frontal areas, and so on?
Well, probably because we see this kind of behavior in all kinds of mammals. You don't have to have a whole big frontal cortex for you to show this deficit, right? So Andy King has done a bunch of really elegant studies where he's shown that those simple interpretations from the psychophysics from the 70s and 80s doesn't really hold in ferrets and probably in any real animal. If you have long enough stimuli and allow the animal to move its head, as was being discussed, And then you can lesion A1, and there's not really much of a deficit. You have to lesion more than just A1 for that to not work. What's interesting in his studies that I find really remarkable is if you lesion A1 in a ferret, and then he's not able, he can overcome, this deficit. But then if you plug one ear, a normal ferret will adjust to that and be able to localize fine, but not one with an A1 lesion. Okay, so A1 is necessary for you to learn how to use these new cues, right? And presumably a place like CL in the macaque monkey is where you're really doing the sound localization, right? And it's not necessarily A1 specific, but it is cortically mediated.
Yeah, because in that sense, in the macaque brain, you have compared quite a number of areas to see where would you find the specificity to the location of the sound source. And it was actually surprisingly specific. Only one area jumped out that really gave you the strongest location modulated response, and actually A1 wasn't one of them.
Right.
So how do you explain that?
Well, the thinking is that the stimulus comes into A1. A1 contains all of the necessary information, and then it projects to CL, and the CL neurons are specific in the way that they interpret that information, and they get the nice small receptive fields, which allows then some other area, not CL, to make some sense out of that and give the monkey the percept of where the sound actually came from.
So, but area CL, if that gives you the percept, is then CL talking to another area to translate that to a goal-oriented action?
Presumably, that's how it works, yeah.
Okay,
probably the caudal parabelt is going to be critical. I imagine that if I did my experiments there, I would see very good correlations as well.
Mm-hmm. But then, what's the latency of the response in CL?
Normally, if you're not old, it's a little bit longer.
Mm-hmm.
And so Joseph Rauschecker did a really nice study quite some time ago, and what he found was that if you lesion A1, the responses in CM, which is its neighbor, to tones goes away, but to noise stays. Mm-hmm. So the thinking is that there's both noise and tone information coming from the thalamus and the cortex to CL, and it's getting combined. And if you take it away, the tone information is lost.
Okay, right? But now the stimuli you use were largely white noise bursts.
Yeah.
Okay. So I could argue then from the perspective of having an auditory percept, that's rather reduced. White noise is much, right? Yeah. So if we now would start to use, let's say, calls of other monkeys or sounds of predators, would you expect that the result might look different, that you might find more specificity in other areas?
More specificity for the different call, for example?
Because now we have meaningful sounds. Actually, you use a sound that is not meaningful.
Right. Well, it was meaningful in the sense that you had to figure out where it was coming from to get
you got a reward to it as well.
You got a reward, yeah.
Right.
So I made it meaningful.
Exactly. Yeah. Okay, so that might have helped then in that sense. Yeah. Okay, you're right. Exactly. So the other thing you then, so now we have localization. We are looking at issues of adult cortical plasticity. Now, the famous experiments people would do with, let's say, the orienting system of the superciliculus, you put prisms on the owl monkey on barn owls, and then you can show that sort of the maps get dramatically reorganized, also in adults, still get the orienting responses aligned. Would you expect to see something comparable in your monkeys when we start to sort of distort this mapping of sound to location? Would you also see similar remappings in CL?
Oh, I predict that you would. Mm-hmm. Right. As soon as the monkey was able to do well at localizing it, my prediction would be that CL would show changes that would be really interesting to look at, because it's not clear. Since there's no topography, you can't really see, these cells changed where their best direction was, right? You wouldn't be able to see that, but you would see that at the end of the day, they all changed appropriately for that to happen. Yeah.
Right.
Yeah.
But now to trigger the goal-oriented movement, do we see something similar as we see in vision? Like in vision, you might go to frontal eye field and then sort of communicate with the colliculus to then set a gaze direction. Would you believe it's a similar pathway that would trigger the orienting response to auditory stimulus and then the movement to the source?
I would imagine that would be the case, but it might be very much different than that.
Why is that?
Well, the visual system and gaze shifts are all dependent on what you see is depending on where your gaze is, right? And so in the auditory system, that's not quite as much. So you saw the psychophysical results for these pretty, fairly not quiet sounds. It didn't really matter where the sound was coming from. You could tell where it was, right? So that would be gaze independent, right? So if you had to orient through the spirochelicus, how it wouldn't surprise me if that was a little bit or a lot different than the visual system, just for that reason, that the gaze shifting isn't part of the sensory input. Right.
True. But the other thing is also for the auditory stimuli, you might have more ambiguity because the visual scene is in front of you and you deal with it, and it's all the information is out there.
Right.
Well, in an auditory display, the source localization might be more difficult, that you would have to make several orienting movements before you really can pinpoint the source.
Right. So the amount of error that you will make I didn't show these data, but if you're in a dark, you hear a sound and I ask you to point your head toward the sound, the amount of error you make is considerable.
Mm-hmm.
Right.
So that means it might be more an iterative process. You do several orienting responses before you really have targeted the source.
Right.
Would it mean that areas like CL have more of a working memory property than you might find in more vision-oriented cortical systems?
That's a good question, and the jury is out on that because the auditory stimuli are quite a bit different than what you see with visual stimuli, and because they're temporary, you don't have really a lasting auditory stimulus. Right? It's pretty rare that in nature that something just hums for a long period of time. Whereas if you look at the rock's there and the rock doesn't move and you see the rock the whole time.
Right, exactly.
As opposed to, there's debate, what's an auditory object? Does it even exist or is it an event? Right? And maybe an event is a better way to think about it. So the way I think about it, what's the auditory system in a primate really supposed to do? You're minding your own business. You hear a twig snap behind you. You can't see what made the twig snap, right? But you hear it and it's a discrete event. And so what do? You orient and you have to get within plus or minus 15 degrees of it. And then you use your visual system to see what snapped the twig. Is it something I can eat, something that's going to eat me, or something doesn't matter, right? So it's really not that common in a primate like a macaque who's diurnal and can use its visual system a lot to really worry that much about what it's listening to. An owl monkey might be very different because it's nocturnal, right? So it doesn't see that well. And maybe in that species, these kinds of things would be quite a bit different. Right.
So then as an application domain of your understanding of adult plasticity in auditory processing, you also start to look at the aging brain.
Yes.
Right. And how the aging brain actually starts to change its responses to auditory stimuli. So what are the most remarkable differences you found there?
Well, I was really surprised when I did those studies that the activity of the brain is so much greater than it is in younger animals, because I came in with the completely false assumption that the older animals would have slower brains that would be less active. I thought they would probably be more like an anesthetized rat brain as opposed to a awake behaving monkey brain, which is very active. And everything makes sense when you're recording from MT in a macaque because the motion goes in a certain direction, the cell screams like crazy, and you don't have to be a neuroscientist to go, I bet it's doing something with the direction of motion, right? It's pretty clean. But the old monkeys, they were more active and they were much more sloppy, right? And the older monkeys that we were studying at the time acted very old. They would sleep a lot. They didn't seem that astute. They didn't seem like they were really perceiving things very well. And I naturally equated that with not much brain activity, right? As opposed to the opposite. So it was really surprising to see that. And when we were doing the studies, we were double checking and triple checking and trying to make sure that we're doing everything right, that we're not messing up with our window discriminators and we're not recording a bunch of noise, and we're always looking for 60 cycle and all these kinds of things to convince ourselves that no, the neurons are really firing more. They're just not firing well, right? So you get this loud, sloppy, ugly signal, which I guess the monkeys themselves means they have, weaker percepts, right? And then that's kind of how it all ends up to work.
But is it more, like, say, a noisy brain? Is there more noise in that brain? Or is it more dynamically modulated, that also if you present a stimulus, the response is still specifically tuned in time, but it's just much much amplified.
It's more amplified, and it's not as specifically tuned in time.
Yeah. So one of the things you mentioned there was the possibility that there was perhaps selective loss of inhibitory systems, which we know are important for tuning up the dynamics of perceptual systems so that you get a clean percept. So I think thinking, for instance, of work in rat S1, Dan Simons, for instance, has talked about the role of interneurons in, shutting down some of the activity that you will get through the excitatory networks in cortex. And then you have a wave of inhibition coming in to make sure that doesn't get out of control. So that it sounds quite consistent with a network which is got some damage, but it's not less activity, as you say. It's just a lack of appropriate tuning of the inhibition systems.
Exactly. So I think what's happening in these older animals is their inhibitory network has crashed, essentially, right? It's not doing as well as it. And that's not just in the cortex. It's in the cochlear nucleus and the spiralive and the inferior colliculus and the geniculate. So it's all along the ascending auditory system that there are these problems. And by the time it gets to cortex, it hasn't been very well refined, right? And so it's a louder, sloppier system, and it just doesn't get any better at the cortical level.
And has anybody looked for sort of selected loss of inhibitory?
Not in the cortex. It's been seen in the brainstem and the midbrain. Donald Caspery's group has shown these kinds of things. And there's all kinds of neurochemical changes that happen down there. And it hasn't been studied, certainly not in the macaque auditory cortex. And the inferior colliculus is the place most people stop on the way up, right? So the cortex is very complicated, and it's got layers and all these kinds of things. So it hasn't been studied to my knowledge.
And I think, as you also said, that there's an implication of this, which is that if someone's suffering hearing loss, the last thing you want to do is actually shout at them because they're overstimulated anyway. So,
slower and lower is the way to go, right? And the other key is if you have a hard time temporally processing, speech sounds are temporally very complex. And words the way we talk, we don't pause between words. We pause at the stop consonants, right? So if you're stopping at only the stop consonants and you're running words together, right, which we always do, and we can interpret that easily enough, it's hard if you can't hear the modulation, right?
Yeah.
So the key to talking to an older person is not necessarily talking louder. That's not going to really work, but to pause between words, right? And that way they can get each one easily. It's the same thing as when you learn a language and you're not very good at it, and then you go to the country and everybody talks really fast, right? Because you're doing word, right? And they're pausing at the stop consonant. So all these words run together, right? Right. So it's same things happening in the older people.
But now, if you look at these the data you present on the response to these older monkey brains versus the younger monkey brains, it's actually the response latency in the older brains seems shorter as well.
Very much so.
Is that correct?
Yes, absolutely.
So, but also the gain is just up in the system.
Right.
So how about an alternative could be that you say, okay, the cochlea, given also that it's partly mechanical sensor with the hair cells moving, that sort of loses sensitivity. And what the brain now must do in response is just crank up the gain in everything that follows. Right? So then it's not so much that we lose inhibition as due to aging, but we're cranking up the gain because the periphery is less sensitive. Would you buy that?
That certainly would be a reasonable possibility, but I think the evidence certainly from the rodent suggests that what's happening is you have less input coming out of the cochlea, and so you adapt to that by doing things that effectively decrease inhibition as opposed to increase the signal specifically. I think that's where the evidence is headed towards at this point. And that's based a lot on neurochemical evidence and the changes in the GABAergic neurons.
Yeah, but there are two ways to think about this, right? Because you can think about inhibition as gain control.
Sure.
Right? Or you can think about it as sort of sharpening a response that you say, look, I have a bunch of frequencies coming in. I set up some sort of winner take all among them. So I sharpen the most salient aspect of my input signal.
Right.
But these are two different views on how you can think about the inhibition. So do you see it as a sort of a non-specific regulatory adjustment or loss, or a more specific one?
If I had to guess, which you're making me guess.
Yeah,
right?
yeah, of course.
So my guess is that it's a little bit of both, but it's probably 70-30, and 70% is the sharpening part, and the 30% is just kind of a global background to let the signal come through.
And that means, in your opinion, this would not be happening at cortex. This would be all subcortical.
I think that's what's happening subcortical, and I'm really keen to look in the cortex and see what... And the cortex is much more well-behaved as far as things like palvarbumin being in GABAergic neurons and things like that. And the brainstem, it doesn't really do that, right? So it would be keen to look in the cortex and see, is there in fact losses of GABAergic neurons, and which kinds, and how would that work out, right? Because there is a general loss of neurons in the cortex of these animals.
And another piece of evidence, I think, that speaks to the inhibitory story rather than this amplification story is the sort of oscillation that you see, where the firing is high and then it drops and then it comes back high and then drops again. And that suggests some alteration in the cortical dynamics rather than just a raising of the gain generally. And that could easily be consistent with loss of inhibitory neurons. So, for instance, you've got fast spiking inhibitory neurons which are coming into the cortex quite soon after the initial excitatory burst. And people are suggesting that these are there to damp down the response and make sure that the neurons that are responding are the ones which are most finely tuned, which would be very consistent with what you're saying. And if that's lost or that's delayed, and you'd imagine dynamics could be upset so that you get these oscillations.
You buy that, Greg?
No, that works for me.
Okay.
That's
because then I think you're both wrong. Because I think if Tony,
the model.
Exactly, if Tony's interpretation is correct, that would also imply a loss of specificity at the cortical level, because I'm tuning down my inhibition at the cortical level, which means if I have overlaps in my receptor field responses, I lose my ability to filter those out. And if I look at your data, you don't necessarily look at the specificity. The specificity is not necessarily lost in the response. It's just really the amplitude that is strongly affected. So I think you're both wrong if you agree with Tony.
Well, I would say if you look in CL, it's pretty clear that the specificity is lost because the spatial tuning is about...
So I'm sunk now, you're saying.
Yeah. I'm sure we're both right.
But there's something else that now worries me because, okay, here we go. We're getting older. True for all of us. Things getting messed up from my cochlea all the way up to my cortex. I get... The responses get strongly amplified, they get much stronger. But in the meantime, you're telling me we have this sort of continuously plastic cortex. So if I'm pumping in higher amplitude signals, why is the cortex not adjusting to this? It's almost like sewing together two fingers, right? I get a stronger drive onto a certain cluster. I have to reorganize my map. So are these maps reorganizing, and are they reorganizing then in a functionally related way?
Right. So how do you put these two things together? How come the old monkey doesn't have a plastic brain to adapt to all this? I don't know the answer to that, but I can guess again. And the way I see what happens is you're in your 40s and your hair cells and your spiral ganglion cells, etc., are starting not to work as well as they used to. And so your brainstem and auditory midbrain is beginning to try to adjust to this. And it's adjusting this by messing around with the inhibitory system. And your cortex is getting a weird signal and it's being plastic and it's changing. And you have no symptoms. Okay, so it works. It's been working for a while, right? By the time you're 78, 79 years old, right, there, for whatever reason, either loss of neurons or loss of the ability to quickly adapt, or the fact that you are slowing down quite a bit and you're not re-updating your maps as often as you are, makes it so that process breaks down. And then you start to do plasticity that's hurting you, right? So what happens when you're a little bit older and you're having a conversation and you keep making up parts of the words that you're missing, and then your model of the conversation, it goes south, and you ask a really stupid question and you get laughed at, right? And how is it as you're older and it's a little bit harder to hear, so you have to, do this, you start to not go into those situations, right? So all of these things that you're normally doing to make sure that your maps are okay and you're doing all right, you start to self-deprive these things. And what's that going to do? That's going to drive your maps in a bad direction, right? So use it or lose it, is what's been said. So if you start to avoid social situations because you can't hear very well, you're not going to get better at hearing.
Sure.
Right.
Well, that's why I'm training myself to not be bothered by posing a stupid question. It's inoculation against my future hearing loss.
But this does also suggest a potential way of developing a program to reduce hearing loss. Is some hearing training that might help us preserve our hearing as we get older. So,
well, that's in some sort what Mike is also doing with his company, Science, right?
Right. So he's doing that. Nina Dronkers in Chicago is doing this with music. Right. Okay. So if you use motor input and then she's looking at the inferior colliculus, but there's no reason it's not being translated throughout the system. And that will give you the benefit of keeping your maps up to date and maintaining that standard.
So if I listen to music, that's going to help me maintain a good map.
It's better than not listening to music, but not as good as playing music.
Oh, even
it depends on the amplitude.
The amplitude?
At which you play the music.
If you play it too loud, that's detrimental. You're right. Yeah.
Well, we can listen to Tony playing this evening. He knows the whole Beatles songbook by heart.
Be too loud.
Okay. But now, so this is really great that you also start to touch upon these more applied issues and try to get insight in how we actually can deal with hearing loss. But on the other hand, you could also argue that the disinhibition we might see with aging might actually also have a positive effect in cognition. We could call it wisdom in some sense, right? Because if people get more disinhibited or more disconnected from, let's say, their immediate emotional responses to things, they can exist in a state of pure cognition,
right,
or not,
or fantasy,
or pure delusion.
Yeah, exactly. Delusion. Yeah.
So now, you finished up by speculations about the homunculus, right? And with also sort of to think a bit about this question, okay, if we have this dot plasticity, we have some handle on its mechanisms, how would you actually be able to grow a new module into a cortex or remove one, right? So how can we do that? How can we add, let's say, a radar map to our own cortex?
Right. So what got me thinking about that was the difference in the somatosensory cortex between Old World and New World monkeys, where Old World monkeys have a clear area 2, which is non-cutaneous, and it's between area 1, which is cutaneous, and area 5, which is visual and non-cutaneous as well. And New World monkeys don't have that so much, right? So the experiments by Jeff Padberg really opened my eyes that there was a big difference, and it always was curious to me that how would you actually make a new field? Because that's what evolution does. We make new fields, and humans have more than macaques, and the common ancestor had some number less than both of us, right? And so the fact that when you train a monkey to pay attention or to discriminate a tactile stimulus on their finger, and you essentially change the morphology and the functionality of an entire cortical area over that representation, area 3A, leads me to think that, well, that can happen pretty quickly, right? And so what would it take to get a new cortical field? So if you wanted a new cortical field, you can either change it in a lifetime, in weeks, but you lose whatever it used to do because your brain doesn't get bigger because it's stuck in the skull, right? So one way that you could do this is you could alter your functional cortical topography by changing your niche, right? And then when you get the opportunity to get a bigger brain, you have that, and then that same kind of process can fill in that space, right? So it just seems to me that you don't have to wait 30 million years to hope that your progenitor cells last a little bit longer so that you get a bigger sheet. Then you decide, what am I going to do with it, right? You can jumpstart that and already have in place something that's new, right? And then when you do get more tissue, you're ready to fill it in with that. Right? So that just made sense to me.
Okay. But now, isn't the notion of homunculus not overrated to start with? Like for instance, this very literal, almost like a copy of the body in the cortex. Also, you already showed this incredible individual variability, right? In how you organize your somatotopic maps moreover, you also within the somatotopic region, you might have, let's say, duplications of such a map. You might have submaps within maps. How should we think really about this? How accurate is something like a homunculus?
Well, it depends on how you measure it. So if you're Penfield and Rasmussen and you put your stimulator every 7 or 10 or 12 millimeters across, you get a homunculus.
Mm-hmm.
Okay. Yeah. If you take a microelectrode and you penetrate every 50 to 75 microns, it's not super duper clean, right? The map of the hand in the owl monkey is, except that the hairy skin is in all kinds of different weird places, and it's, there's overlap, and it looks pretty good. Other parts of the body, not so much, right? So when the receptive fields start to get big, like on the rump or the leg, it starts to get a little bit sloppy too, right?
So now to finish up our debate here, or our conversation. So you started out to look at this issue of adult plasticity. We're at the beginning, so actually getting this back on the map again, right? Which is great. And to travel all the way through, let's say, somatosensory systems, motor systems, auditory systems. So now, if we really want to follow that tradition in which you have been working, what is Gregg's law that we should adhere to understand the brain?
Gregg's law to understand the brain.
Mm-hmm.
It's harder than it looks. It must be harder than it seems. Yeah. Yeah. Well, I think if you want to understand the cerebral cortex, and this is what I've done, so that's why I think it, you should not get stuck in a cortical area. Okay. You should not say, I want us to understand how the cerebral cortex operates to provide perceptions. And to do that, I'm going to spend 30 years studying MT. Mm-hmm. Okay. So I think you're not going to gain the insights that you need unless you look across a bunch of different cortical areas. Right. And, a bunch of different species would probably be pretty smart too. But I'm not only a cortical snob, I'm a primate snob too. Mm-hmm. So I've been sticking to the primate. But one nice thing about being a close colleague of Leah Kribitzer's is that she studies all kinds of different animals. And so I know all about these things, and it's something that you can really use to gain your insight. So if you're not going to study them yourself, you should at least bone up on the literature and talk to somebody who does, right, so they can keep you grounded.
So embrace variability would be it.
That would be a good one. Yeah.
Okay. So 5 years from now, Tony likes traveling. He comes visit you in Davis, and he's going to check whether you verified the hypothesis that you're going to share with us now. So what's the most important hypothesis you want to see verified in that time frame, 5 years?
In a time frame of 5 years?
Yeah.
So that's, in the monkey world, that's not very much time whatsoever, right? So what I would like to see is how is it that this proposed parallel pathway with the rostral and the caudal wear and the rostral wet pathway, what is that really like? I mean, how is it really doing? My money is that it's not going to be quite as clean as it is in the visual system, but it's going to probably provide us a lot more insights, I hope anyway, into how not just the visual system, but also the somatosensory system and others, how you actually do the binding of these objects into, a single kind of thing. So I'm hopeful that 5 years from now, when Tony comes by, I'll be able to tell him almost how it works.
Great. Gregg Recanzone, thank you very much for this conversation.
Thank you.
Thank you.
The CSN podcast was produced by the Convergent Science Network of Biomimetics and Biohybrid Systems, a project funded by the European 7th Research Framework Programme. For more interviews, recorded lectures, or upcoming conferences in the field of biomimetics and biohybrid systems, go to csnnetwork.eu. And thank you for listening.
You're off the hook.
All right. Well, thank you, guys. That was never done that before.
I thought it was cool.
Yeah, it's good.
I like it.
Yeah.
Nice. Thank you.
You're welcome.
It's great. I didn't know you were instructed by Leah to talk about this.
Yeah.
If Leah wouldn't have given you any instruction, what would you have talked about?
Probably I would have talked all about the auditory cortex.
Okay.
That's all I would have talked about.
Right.
And I would have done the, we also looked at a whole bunch of different things in the temporal domain. Right. So I would have talked about that.
Ah, cool. Okay. I didn't know that.
Yeah.
But all cortical.
All cortical.
Okay, what's the time window in which cortex can reliably represent interval information on its own?
Like how fast can it?
No. If I give you a beep, let's say, or you have to hold a stimulus in memory for a certain amount of time, what's the time window in which cortex can do this reliably in a task?
The time window in which a monkey can reliably do, say, a delayed match to non-sampler,
for instance,
that, seconds.
Seconds. And where is the information stored? Is it reverberating in the cortical circuit, or?
That's a question you'd have to ask one of 5 or 6 of Mort Michigan's postdocs that couldn't figure it out.
But do you have any idea? Is it the cerebellum or is it cortex?
It would probably have to be, I think it would probably be in the cerebellum. It gets to the auditory system gets to the thing that I said before. I can hold this in my visual working memory really well, right? But the auditory system doesn't have objects like this that you have to do. And so it's been a real difficult thing for the field to figure out what you can reliably remember in memory. So you can train a monkey to listen to a bunch of sounds, and when it hears boop, boop, beep, to let go. And it can remember that all day, and that'll be held in there forever. But to do the classic beep, 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 okay, now is it beep, boop, beep, or beep, beep, boop? And it's like, I can't remember, and so all of those, Mort tried for years to do those ablation behavior experiments that made him famous in the auditory domain, and just couldn't get the stupid monkeys to. I mean, he started those when I was in Mort's lab.
Right.
Yeah.
So you play guitar?